gathering, I am pleased to witness such a varied and involved group of participants. Your presence contributes greatly to the depth of our discussions and highlights our shared dedication to progression in our respective areas of expertise. I look forward to fruitful interactions and mutual learning as we embark on this educational journey. SPSR is acclaimed for its engaging webinars, further establishing itself as a leading platform for continued professional growth within the pharmaceutical field. I am certain that today's webinar will uphold our tradition of excellence, presenting valuable perspective from a distinguished presenter. Let's embrace this chance to gain fresh knowledge, participate actively, and move our industry forward. Welcome to everyone, and I look forward to a beneficial experience for all participating. Thank you for your presence. We are gathered today to commemorate the 59th SPSR webinar on Translational Phytomedicine for Cancer, Bridging Laboratory Research and Clinical Applications, a distinguished event in this series hosted by the Society of Pharmaceutical Sciences and Research. Under the esteemed guidance of Dr. Shashi Alok, the National President of SPSR, Society continues to play a pivotal role in fostering innovation and advancement in the field of pharmaceutical sciences. The SPSR is continued to the progression and enhancement of the pharmaceutical field through diverse strategies. We aim to facilitate conferences that promote pharmacy-related initiatives and endorse programs and activities addressing public health issues in India, with an emphasis on disease prevention. We also make it a point to acknowledge and celebrate pharmacists whose significant contributions inspire further advancement in our field. Central to our purpose is the commitment to fostering the growth and spread of pharmaceutical knowledge, ensuring a consistent and rich exchange of information regarding pharmaceutical practices and services. By pursuing these goals, we strive to raise both the quality and influence of the pharmacy profession, cultivating a proactive and cooperative community. It is our great privilege to introduce Dr. Lekshmi Nath, Associate Professor, Department of Pharmacognosy, Amrita School of Pharmacy, Amrita Vishwa Vidyapeetham, Kochi Campus, Kerala, India, as a distinguished speaker. Dr. Lekshmi Nath's expertise greatly contributes to our event, providing substantial knowledge and insights. Her participation is certain to enrich our discussions and expand exploration of traditional phytomedicine for cancer. We are grateful for her engagement and anticipate an enlightening session. It is our privilege to introduce Mrs. Monika Sabharwal as the convener of today's webinar, who also serves as the National Secretary of SPSR and Managing Editor of both IJPSR and IJP. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of her commitment to the field. We are very grateful for her guidance in today's session. I am pleased to invite Mrs. Monika Sabharwal to warmly welcome our distinguished guest speaker, Dr. Lekshmi Nath. Over to you, ma'am.
Thank you so much, Dr. Pradeep Gulani. Hello, everyone. I am Monika Sabharwal. It is my pleasure to welcome you all to the 59th SPSR webinar on Translational Phytomedicine for Cancer, Bridging Laboratory Research and Clinical Application. This event, organized by Society of Pharmaceutical Sciences and Research, SPSR, and we are privileged to have such an overwhelming response with over 2,500 registrations from more than 20 countries. Today, we are honored to have with us Dr. Lekshmi R. Nath, an esteemed associate professor in the Department of Pharmacognosy at Amrita School of Pharmacy, Amrita Vishwa Vidyapeetham, Kochi Campus, Kerala, India. Dr. Lekshmi is a distinguished researcher whose groundbreaking work in the phytomedicine and the liver cancer has garnered international recognition. We look forward to her insights on the very crucial topic of bridging laboratory research and the clinical application in the cancer treatment. The focus of today's webinar, Transitional Phytomedicine for Cancer, is particularly pertinent in the current era of integrative medicine. phytomedicine, the use of plant-derived compounds for the therapeutic purposes, holds a great promise in fight against the cancer. However, the journey from laboratory research to the clinical application involves numerous challenges and complexities. This webinar aims to shed a light on the latest advancements, strategies, and collaborative efforts required to translate these promising research findings into effective clinical treatments. As we celebrate the 59th webinar in our series, I would like to take a moment to reflect on our journey. The support we have received from around the globe has nothing a sort of incredible. Your continuous feedbacks through the various platforms has been tremendous to boost our team, affirming the importance of our efforts. Before we begin, I would like to extend a very warm welcome to Dr. Shashi Alu, our National President, whose visionary leadership has been pivotal in the remarkable transformation of the SPSR. The SPSR team has been working tirelessly to uplift the pharmacy profession, and it is heartening to see our audience dedication in regularly attending these webinars, particularly on Sundays. Your commitment not only inspires us, but also drives us to strive for the excellence in every session we organize for all of you. Our goal with these webinars is to provide a platform for knowledge sharing and professional growth. And the response we have received thus far motivate us to keep pushing forward. We are committed to make each webinar better than the last one, ensuring that we deliver a valuable content that make a positive impact on our profession. I would like to extend heartfelt thanks for the significant contributions made by the Dr. Pradeep Golani, the co-convener of this webinar, alongside the organizing committee members, Dr. Sanjay Nadev, Mr. Ankur Agarwal, Mr. Junaid Tantre, and all SPSR team members. Their relentless dedication has been instrumental in bringing the vision of this event into a future. The seamless execution of this webinar is a testament to outstanding efforts of our team working behind the scenes. Their dedication to high quality work and the persistent effort have been pivotal to the success of every event we organize. My gratitude goes to each member of SPSR team for their careful management of the details and enduring enthusiasm that brought this webinar a future on. Stay ahead in the field of innovation with the SPSR. Join our YouTube channel for the unique webinars, expert perspectives, and the recent findings in the field of pharmaceutical sciences. Engage with a vibrant community that is involved in the conversation that will progress the healthcare. Keep up to date by subscribing to our SPSR YouTube channel and turning the notification bell icon for the immediate updates. Follow our progress in advancing the pharmaceutical sciences that will benefit the society. Once again, thank you for joining us today and being part of this remarkable journey. Let us look forward to an engaging and enlightening session with Dr. Lekshmi Nath ma'am. Thank you for joining us today and for your continuous support of SPSR's initiatives. Over to you, Dr. Pradeep Golani.
Thank you, ma'am. I appreciate Mrs. Monika Sabharwal for granting me the opportunity to present this webinar. And now we will start with a short introduction of guest profile. Dr. Lakshmi Arnath is an accomplished associate professor in the Department of Pharmacognosy at the Amrita School of Pharmacy, Amrita Vishwa Vidyapeetham, India. With a profound expertise in phytomedicine and liver cancer research, Dr. Lakshmi has made significant strides in the scientific community, particularly through her groundbreaking work on hepatocellular carcinoma. Dr. Lakshmi completed her PhD in the Cancer Research Program at the Rajiv Gandhi Center for Biotechnology, Trivandrum, Kerala, an esteemed autonomous research institution under the Department of Biotechnology, Government of India. Her pioneering PhD research on hepatocellular carcinoma led to 4 international patents in US, Canada, Japan, and South Korea for her innovative work on Eutrocyte B and derivatives thereof as therapeutic for hepatocellular carcinoma. This remarkable achievement earned Eutrocyte B an orphan drug designation from the US FDA, underscoring its potential as the chemotherapeutic agent against liver cancer. In a significant advancement for cancer treatment, RGCB signed an MOU with the Oklahoma Medical Research Foundation USA to facilitate the clinical translation of Eutrocyte B for liver cancer patients. Additionally, the technology has been transferred to the multinational company Q Biomed, marking a major milestone in bringing this promising therapy to the market. Dr. Lakshmi holds an M.Pharm from KLE University's College of Pharmacy, Karnataka, and a B.Pharm from the Department of Pharmaceutical Sciences, Trivandrum campus, Kottayam. With over 10 years of research experience at RGCB's Division of Cancer Research, she has made the substantial contribution to the field of phytomedicine and liver cancer. She recently edited a book titled Liver Cancer and Phytomedicine, Chemopreventive, Chemosensitization, and Chemotherapy Strategies, published by the World Scientific Publishing Cooperative Private Limited, Singapore. As a thought leader in her field, Dr. Lekshmi serves as the guest editor for prestigious journals such as Frontiers in Oncology, Frontiers signaling events associated with the progression from fatty liver to hepatocellular carcinoma, and the chemotherapy strategies. Additionally, she explores the potential of traditional knowledge in combating various liver disorders and infectious disease, focusing on immunotherapy, investing their mechanism of action, and validating them through preclinical models. Dr. Lakshmi Arnath's extensive expertise, innovative research, and dedication to advancing cancer therapeutics make her a distinguished speaker and a valuable contributor to the scientific and medical communities. It is with great satisfaction and pride that we declare that we have exceeded 2,500 registrants globally, showcasing an impressive diversity of countries. Significant attendance is noted from the nations including India, Bangladesh, Brazil, Bulgaria, Cambodia, China, Ghana, Indonesia, Kenya, Malaysia, Nepal, Nigeria, Oman, Pakistan, Saudi Arabia, Syria, USA, and Uzbekistan. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude is extended to each person who has taken a moment to participate in this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker, Dr. Lakshmi Arnath with us. Welcome, ma'am. Your insights would be highly valuable, and I would appreciate if you could present them in a speech. Over to you, ma'am.
Thank you, sir. Nice introduction. So very warm good morning. So myself, Dr. Lekshmi Arnath from Amrita Vidyapeetham, Kochi campus. So today's topic of presentation is Translational Phytomedicine for Cancer, Bridging Laboratory Research and Clinical Applications. So to begin with the topic, so hallmarks of cancer was proposed as a set of functional capabilities acquired by human cells as they make their way from normalcy to neoplastic growth states. More specifically, the capabilities that are crucial for their ability to form malignant tumors. They currently embody 8 hallmarks, capabilities, and 2 enabling characteristics. So in a normal tissue, somatic mutation sporadically arise. So these mutant clones can still maintain homeostasis until they are composed of additional stimuli. So their proliferation becomes uncontrolled and malignant transformation initiates, progressing from pre-malignant lesions to advanced tumors. During this process, the transformed cells gradually accumulate additional genetic mutation and epigenetic alteration. So so the global cancer burden is growing. So as per WHO guidelines, it is expected to be 35 million new cancer cases by 2020. It's a serious, so we have to be very alert. So cancer is an abnormal state of cells where they undergo uncontrolled proliferation and produce aggressive malignancies that cause millions of deaths everywhere. So many types of cancers are diagnosed in human population, like throat cancer, lung cancer, stomach cancer, bowel cancer, then breast cancer, so liver cancer, etc. So the therapeutic options, n number of options are available, like hormonal therapy, surgery, bone marrow transplantation, chemotherapy, targeted therapy. So the advancement of cancer therapeutics lead to targeted delivery of anti-cancer drugs, personalized medicine, then immunotherapy, etc. So let me draw your attention to the history of chemotherapy. So this begins in the year 1942. So in 1992, FDA approves first blockbuster oncology drug. So we are all familiar, the Taxol. So how many of us know the inventors behind this wonderful drug? So this origin of this wonderful chemotherapeutic drug Taxol is from Monroe and Mansu Bani. So at the Natural Product Laboratory, they discovered and elucidated the structure of Taxol. So not only the Taxol, so they have derived n number of molecules, and camptothecin is also another life-saving compounds for treatment of cancer. So you can see in this picture, this actually this American Chemical Society published in 2003, the discovery of camptothecin and Taxol, the wonderful drugs, the commemorate booklet produced by the National Historic Chemical Landmarks Program. So we have to know about these kind of people, so who made significant contribution, even if they don't come into the light of a Nobel Prize. So we know more than 50% of the natural products, especially the plant-derived products, specifically used in anti-cancer therapy. So let me draw your attention how these discoveries, invention from plant-derived molecules is happening. So have they doing any the preliminary kind of work that may make into an advanced kind of work? Yes. So we need to have a cell culture technique to preliminary screen these molecules. So you are familiar. So if any sort of drug, so for example, if it is a root or leaf or rhizome, so you have to make it into a standardized manner. So whether it has any kind of contamination, so you have to do proper authentication of the plant material. So once you complete that process, you are assuring your raw material selected for your further studies is authenticated and contamination free and properly standardized. Then you can move on to the second stage of extraction. So you can adopt any number of options. Nowadays, microwave assisted extraction, supercritical fluid extractions are also available, especially in industries. But as far as concerning about our usual ordinary laboratory techniques, we'll adopt maceration or Soxhlet apparatus. So once your extract is ready, so next stage will be the screening of the active extract. So you can prepare n number of extract out of a plant part, then rhizomes, roots. So you can either adopt aqueous extraction or organic extraction. So to which extract you will choose, that is a crucial thing. So for that, you have to follow the bioactivity graded process. So you are selecting the most active extract, then subjected to isolation process using open column. So further to extend HPTLC, etc. So once you prepare the extracts or once you collect the fraction from column, the ultimate point is that you have to identify its activity. So for that purpose, we need to perform a preliminary assay known as MTT assay. MTT is most commonly used. Other kind of cell viability assays are also available. For example, SRB assay, etc. So most of the time, if you search 10 papers out of 5 to 6 papers, the preliminary assay will be an MTT assay. So you have prepared that extract, you screen for its cytotoxic activity by MTT assay. So along with that, you can connect a few assays, cell death assays, to further establish, further confirm its cytotoxic anti-cancer effect. So once you have selected fraction, then extracts, or in between you are moving through the compound isolation in collaboration with any chemistry department. So you will get a pure molecule as well. So in which all area you are focusing, so you need to perform an in vivo study along with the toxicological evaluation before moving on to further extensive evaluation of that particular molecule. So for animal study, most commonly for our anti-cancer studies, xenograph models are available. That is an immunocompromised mice model, or else orthotopic models. Orthotopic model, syngeneic model means a specific kind of rat cells can be inoculated directly into the particular area. So if you want to develop a liver cancer, you can inoculate that particular number of cells into that liver, left lobe of liver, so that you can develop a model, so you can test your compounds. So the preliminary assay will give you an evidence about which type of cancer you have to select. So based on the activity, which cancer cell line gives you the best result out of MTT assay, you are choosing that particular model. For example, suppose your extracts, compounds, fraction, so whatever, so they give most active effect on that cell line cervical cancer, you have to choose the cervical cancer. So if it is giving very good effect on HCT cell line, colon cancer cell line, you have to choose the colon cell line. So this is all about the basic concept behind the preliminary studies on anti-cancer compounds. So I'll be talking about a few molecules which I have come across during last 14 years. I started my cancer research at Rajiv Gandhi Center for Biotechnology as a PhD student under Dr. Ruby Jonando. So in the year 2009, so till now, currently I am working as a faculty at Amrita Vidyapeetham, Amrita School of Pharmacy at Department of Pharmacognosy, Kochi campus. So being for the last 14 years, so I have come across a few molecules. So what I feel, so only a few molecules will be translated into a clinical status. So we approach any molecule, we intend to produce it as a clinical utility. But most of the time what happens, only a certain set of molecule can be translated into a clinical setup. So the most interesting molecule, what I found during my last 14 years of research, the foremost thing is Utroside B. So you may have seen this news in newspapers a few years back, maybe 2 years back. So liver cancer drug based on Manitakali compound identify RGCB. So this is my PhD work. So in this particular molecule, what we found, we reported the anti-cancer efficacy of utroside B, which we isolated from Solanum nigrum. This utroside B produced IC50 500 nanomolar, which is 10 times more potent than that of sorafenib, the only FDA-approved drug against liver cancer. This was the specialty of this particular molecule when we come across at the initial stages of our preliminary screening. But we are not sure about at what extent this molecule will move forward to make it into a big success. So I'll tell you the story. So we have published this paper in 2016. So I started my journey in 2009 on this particular work. So I could publish a paper only on 2016. So that is one of the drawback of this kind of plant-based research. Will take a lot of time, but there is a hope always. We have to keep to move forward. That's all. So this is our collaborator. So we are fortunate to get a wonderful collaborator. So that we could elucidate the structure of that particular molecule. So the problem with any kind of the phytochemical research, so as pharmacognostic pharmaceutical background, we could attain at a certain level of isolation. So the chemistry background researchers could help us to elucidate that particular structure if it is of a novel compound. So usually what we do, so if it is already a reported kind of molecule, so we can compare with the literature or compare with the available spectra, we can do the elucidation. But usually for a bioactivity gated isolation, it's not possible because we are chasing an unknown molecule. For that, we need to take the help of a chemistry group to elucidate the structure of the novel molecule. So as I belong to the Biotechnology Institute, so we got a number of opportunity to study the details of this particular compound. So we check the cytotoxicity. Once we check the cytotoxicity, confirm the IC50, we move forward to various apoptotic assays. So apoptotic potential of utroside B evaluated in cell culture system, analyzing various key molecules like PAR, caspase, etc. These are markers of apoptosis. So moving on to the next stage. Once we identify the IC50 value, confirm its cell death mechanism. So the most important thing, so for any molecule to act as a chemotherapeutic agent, it should fulfill two essential criteria. It should be pharmacologically safe and it should effectively induce anti-tumor agent in vivo condition. So keeping this in our mind, so we'll look forward the toxicological evaluation of this utroside molecule. So we performed the acute and chronic toxicity studies, doesn't produce any toxicity, and the histopathological verification was done by the concerned pathologist. So next step was to check the anti-cancer effect of this particular molecule in an in vivo system. So most of the time, this failure in the plant research or any sort of the drug discovery process, so it will produce very good effect in vitro cell line system. But when we study those particular compounds in vivo system, so the result will be unsuccessful most of the time. So what we did here, we conducted an in vivo validation of anti-cancer effect using HepG2 NOD skip mice model, which is an immunocompromised mice, where you can see a flank region. In that flank region, so we can inject the HepG2 cell line, which is a human-derived hepatocellular cell line. So to that point, after a period of time, the tumor will be visible. So we can evaluate easily the tumor reduction volume and the corresponding molecular markers. So for that, we conducted in vivo evaluation of this particular molecule using this immunocompromised mice model. Surprisingly, we didn't see any visible tumors in Utrosid B treated mice from outside. Only when we sacrifice the animal, we could see a very small tumor in Utrosid B treated group. Then further, the tumor collected from that checked for the expression of the apoptosis marker PAR. So we could confirm to a certain extent, so it will produce a significant effect on in vivo system as well. So further to extend, so we studied the molecular mechanism of this particular compound. So most of the time, the chemotherapeutic agents act through induction of apoptosis. So in spite of the induction of apoptosis, as we observed through the microscope for control as well as the Utrosid B treated cells, so we could find small vacuoles kind of formation. So Utrosid B also induces vacuoles. So we confirm that this vacuole formation is an indication of autophagy. It is another kind of cell death mechanism. So further to extend, further to confirm this effect, so sometimes morphological visualization may not be appropriate to confirm a particular cell death effect. So we have to check the further markers to confirm this effect. So we have used an LC3 protein marker, which is an autophagy indicator. It's a classical marker of autophagy. So along with the various autophagy markers like ATG7, Becklin, ATG5, so we could confirm that this molecule not only induce apoptosis, also induce a kind of death program known as autophagy. So when we go through a few literatures, so we could find a certain kind of interaction with this autophagy and apoptosis. So next attempt was to vote whether it is a contradictory or complementary. So to further extend that mechanism, we have done a few set of assays. So to elucidate the autophagic flex mechanism, we used baflomycin, which is an autophagy inhibitor. So conducted a Western blot analysis to confirm the expression of treatment of baflomycin alone and baflomycin in combination with Utrosid B and also Utrosid B. So with all these assays, we could confirm that it induces both apoptosis and autophagy. Only we need to clarify whether what kind of relation between this apoptosis and autophagy. So for that, we have used two major pharmacological markers of autophagy and perform the markers of evaluation of this apoptosis markers like caspase-9 and PAR. So when we combine these autophagy markers, autophagy inhibitors along with Utrosid B, we could find that so inhibition of autophagy is enhancing the apoptosis. That means we could confirm that it's behaving as a autophagy inducer as well as apoptosis inducer, but so at a certain extent, it behaves in a way of autophagy mode of cell death, and after that, it behaves in apoptotic mode of cell death. So as I mentioned earlier, so most of the chemotherapeutic agents act through apoptotic mechanisms. Also, so this autophagy is having a dual role. It's also act as a survival mechanism. So what we expect from our compound is to make it more active, more chemotherapeutically efficacy. So we need to have an inhibition of autophagy further leads to apoptosis mechanism. So we have published this result in Frontiers. The blockade of utrosid B-induced autophagic pro-survival signals augments its chemotherapeutic efficacy against hepatocellular carcinoma. So in the year 2022. So let me draw your attention. So we have published papers, we have filed patent also. So in the year 2016. So for what happens with this molecule. So the main thing is there is only a prime line drug, sorafenib, for hepatocellular carcinoma. So and also in vitro and in vivo condition, it produces a comparable very good effect, and in vitro system approximately 10 times more potent than that of sorafenib. So this company, Q Biomet, a pharmaceutical company, so taken up this molecule and started phase 1 clinical trial in humans. And we have also received a chemotherapeutic utrosid B orphan drug designation for HCC approved by FDA. So you can see n number of news. If you search utrosid B in Google, you can find n number of information, updation on the status of this molecule. So please remember that it started the work in 2009. So still that process is going on. And so we have to understand one fact that so will take time, but it should be useful to the society. So this is also a news published in previous year. So so last year, as a very much this molecule grabbed high attention in the community. So we have received many more than 5 patents for this particular molecule. So moving on to the second molecule, camphoride. This is not a very magical molecule. As usual, it is a simple flavonoid molecule. So, so we have tested in a number of cell lines, but we could get a very good effect in cervical cancer cell line, and we published a paper in RSC Advance in the year 2019. So as I mentioned earlier, for any molecule to be isolated from a plant materials, it has to undergo the laboratory techniques like the preliminary screening by FGT, followed by the apoptotic assay, acridine orange, and annexin staining, caspase assay, PARP assay. Then coming to pharmacological safety, then anti-cancer potential. So this molecule only reached to this extent. So we haven't patented this molecule because most of the time the patent comes, the utility is very important. So when you compare the cervical cancer with that of liver cancer, so we thought liver cancer is having a limited number of drugs. So and also the IC50 produced by the utrosid B was very significant as when compared with the currently used medicine. So we have gone through patent and further procedure, and it has gone in a very good way. But the second molecule of my PhD topic, it went into a paper format only. So here the cervical cancer vaccines are available, and this is a kind of flavonoid molecule. It has a lot of bioavailability issues as well. So we haven't patented this molecule, so we just published this work. So moving on to my independent work process at Amrita. So I left RGCB in 2017, and 2018 I joined Amrita. Then I started working as an independent researcher, and now 7 years completed. So I continued my journey as a researcher in a particular area of hepatocellular carcinoma. So as I belong to Pharmacognosy departments, I am much keen interest to enhance the research associated with the phytomedicine as well. But along with the background of the biotechnology techniques. So we have published a paper on camphoride on hepatocellular carcinoma in 2023. So here the camphoride produce a different IC50 in cervical cancer, as you have seen in the previous slide, and it produce another kind of effect in HCC cell line. So we have tested in different hepatocellular cancer cell line to confirm its effect. Then morphological examination was also done, produced very good apoptotic effect. So I have included very few results into that due to the limited number of slides. Then we have checked the effect using assay kit for caspase 3 and caspase 9. Here in Amrita, what we adopted for this self N1S1 for inducing orthotopic animal model. Previously, I've been using the HEPG2 xenograph model. So here we had a very wonderful animal research facility. So he started using the orthotopic model. So comparing this HEPG2 xenograph model, so orthotopic model, the experiment initiation, validation, everything is a little bit difficult because we are inoculating that particular hepatoma cell line into the left lobe of liver. So in this process, so we could attain a particular cell number to produce the tumor on the left lobe of rat. So the model is known as N1S1 syngeneic rat model. It is a widely established model, but in every institute we have to make it initiate to standardize that condition. So animal to animal, even if the literature and evidences are available, but we have to make it into a condition of in our facility how it works. So the animal standardization is very important. And if when you start doing animal experiment, you can feel the difficulty. So even if you do a particular set of experiment in one institute, the same set of experiment, if you are doing it in another institute, that it will take, even if you are experienced or expertise in that area, it will take a more time. So here we have developed and validated an N1S1 animal model. So we could publish this camphoride work in hepatocellular carcinoma using N1S1 animal model. So moving on to the another molecules. So here the only difference from the camphoride, so another molecule camphorol is see phenolic hydroxyl group at the fourth position of the ring here. So methoxyl group, only that difference. So when it comes into the translational utility, so it had a lot of problem, even if it produced very good effect in vitro and in vivo conditions. So bioavailability is always an issue with this flavonoid compound. So before moving on to address that issue, so we have done a few set of experiment on this particular molecule, how it works, will it be beneficial in hepatocellular carcinoma. So we have published this work as well, the camphorol as an AKT-dependent mTOR regulator, so by targeting FKBP12 in hepatocellular carcinoma in 2021. So along with that, we had a kind of sensitization work with camphorol for liver cancer. So we could produce a very good synergistic effect in animal model as well. So moving on to the next stage. So we have completed all the studies, all the preclinical evidences are appropriate. Still we face many sort of difficulties. So when you search in Google, Science Direct, Scopus, wherever the search engine, you can find n number of molecules, n number of publishing documents regarding the utility of these molecules. But every researcher is stuck into a particular area, like, so it's very difficult to translate this molecule, even if we show it produce very good effect in vivo system. So we need to have a support system from a clinical person to translate these kind of idea and to address the issues faced by our phytomedicine. So we had a wonderful collaborator, Dr. K. Pavitran, so clinical professor, medical oncology, Amrita Hospital. We've been currently sorting the issues associated with this, the current treatment strategy, how we can tackle this drug resistance, that issue, the side effects associated with the treatment strategy in combination with this phytomolecule. So we have a number of formulation system. Is even if the liposome is the most widely used medicine, so we haven't seen, except for Taxol, Docil is available, except for this Taxol-based formulation, other kind of molecules are coming into a kind of good research or good translational potential. So still research is much needed in this particular area to make a revive for this phytomedicine into a mainstream strategy, so that allopathic doctors can suggest that formulation to be utilized in their clinical setting. So I'm happy that I could share a few of my experience as a plant-based researcher, cancer researcher. So this is not a conclusion. So it's just like a take-home message. So plant molecules can also be used as a monotherapy aspects, provided we address its bioavailability issues. And also this phytomolecule is wonderful in addressing the side effects caused by the chemotherapeutic agents, the resistance by chemotherapeutic agents. But still our problem is, so we have to have a well-designed, validated clinical evidence based on addressing the bioavailability issues. So I take this opportunity to acknowledge my humble pranams to Amma, our Chancellor, our inspirations. I thank Dr. Shantikumar V. Nair, our Dean of Research, Dr. Sabita M., Principal, School of Pharmacy, and my mentor, Dr. Ruby John Ander. So we had a wonderful research group exclusively working on liver cancer and phytomedicine. Hope will achieve a certain target of clinical translation in near future. Thank you.
Thank you so much, Dr. Lakshmi, for presenting a wonderful session on translational phytomedicine for cancer, bridging laboratory research and clinical applications. You started the session introducing us about the hallmark of cancer and multi-stage tumorigenesis, different types of cancer, and remedies for treatment of cancer. After that, you discussed about the wonderful chemotherapeutic drug, Taxol. Then you focused your talk on chemotherapeutic agents from plant source, their extraction, bioactivity assays, and animal studies. After that, you introduced us about the research of your anti-cancer drug, Utrocyte B, useful in treatment of liver cancer. You explain how Utrocyte B induces apoptosis, mode of cell death in liver cancer cells, and Q Biomed company planned a phase 1 clinical trial for this drug. After that, you focused your about your research on camphoride, a flavonoid drug that induces apoptosis in cervical cancer cells. Thank you so much, ma'am, for educating all of us about these molecules on which you have done researches. And after that, there are some questions from the participants. I'll be asking you one by one.
Sure.
So, first one is, ma'am, how important is multidisciplinary collaboration in the field of translational phytomedicine and how it can be fostered?
So my experience as a PhD student, so even if I done one PhD with RGC Rajiv Gandhi Center for Biotechnology, we had a facility of biotechnology related work. But we had the lack of the chemical research group exclusively working on the spectral analysis, structural elucidation, etc. So for any work to be into a fruitful translational state, so so no one is so will be expertise with all sort of work. So only a certain person will be expertise in one area, and another person will be in a different area. So as this phytomedicine is exclusively a kind of interdisciplinary work, we need to have a person from biotechnology background, chemistry background, exclusively these two background. Along with that, if you can incorporate a person belong to a kinetics group, pharmacokinetics group, so it is wonderful because we can address the bioavailability issues. So also if you include a pharmaceutical chemistry person in that, you can include any derivatization which will make that, which will sort out the bioavailability issue. That is also a wonderful thing. So multidisciplinary approach is very, very important to make it into a translational aspect. So no one can
Right.
sit in a room and do their work exclusively for them. So after all, what we are doing now, we make it useful for society. So it's better for even if you work with any small work, so better you explore with other sort of researchers because they may have another kind of expertise. So you can save that time. So if you start doing that work and along with your current work, it may take another 10 years. So better you collaborate because nowadays we had the MOU, like we can sign an MOU so we can equally contribute. There is a document for that. So you can have a number of opportunity. And the second part is how to foster the collaboration. Right. So, so I would tell you, like collaboration may not happen within a day. So we have to slowly we have to make it into a progress. Like, for example, you can attend any workshop, then conferences, you can meet people, you can present your own work. Even if it is a small kind of work, you have to present in any community. So at least if you attend a few session or few years of conferences, you may have contact and you will be into that sector. So only that these sort of activities may enhance our knowledge. Like, so we can enhance our knowledge through our through our papers, reading regular read of articles and everything. But we can have a good kind of interaction with other people who are expert in a specific kind of area. So that won't happen in a day, will take time.
Right. Thank you so much, ma'am. The next one is, ma'am, what role do preclinical models play in the development of phytomedicine for cancer treatment, and what are the limitations for these models?
So for a cancer research model, you can classify either as a carcinogenesis model. Carcinogenesis model means you can induce any carcinogen. For example, in the case of liver cancer, aflatoxin. This aflatoxin, if you give animal for a certain period of time, the tumor will develop inside. So that is a long duration. And also the animal used for that particular aflatoxin model will be a kind of days animal, mice. So these kind of models are very expensive and time consuming, but very valuable kind of model. So one of the easiest model I would suggest you, like HEPG2 xenograph model. Even if the maintenance of a immunocompromised mice is very expensive, but at least you can see that tumor developed on the flank region where you have injected that particular cell line. So that if you have a control animal and a treatment animal, so if you are injecting that tumor, cell HepG2 cell line, tumor cell line into that particular flank region. So after 2 weeks, the tumor will be developed on that particular area. Then simultaneously, the kind of treatment groups are also there where you can easily see that tumor, that difference, the control versus treatment. So, but the maintenance of this animal is very expensive because it is an immunocompromised. It need a separate animal facility. Sometimes we have to import also that animals. From Jack's laboratory. Or else another model like N1 S1 rat model, so what we can do that we can inject the cells and develop into an orthotopic region, but the limitation is 50% of aggression. Sometimes if you develop that animal, 4 animal, that particular number of cell line, so only 2 animal may develop the tumor. So that too not visible. Actually, it is an orthotopic region because the inoculation is happening inside the liver. So you need to have about like MRI, CT, etc. to visualize the tumor into that particular region. And also the regression also will happen. Sometimes you may not trace out whether it is due to the effect of your drug or due to that regression that tumor is reduced or it's gone. So every model is having its own limitation, but we have to use 1 or 2 model to standardize that particular kind of effect. So that will take much time. That's why the cancer biology research is not very much happening in our regular academic laboratories, animal facilities, because it is very expensive and also we need manpower and we need much attention. So in a NCA kind of national institute, what happens? No, they have facility for each thing. So they no need to give much attention to everybody because they are established one. But still, I'm saying, but with the time being, we can have, if we work slowly, we can move to that extent. So we need to have a kind of patience into that and sincere efforts. And for everything is, we need to have a passion for that. That's all.
Thank you so much, ma'am. The next one is, ma'am, what are the regulatory challenges faced during the clinical translation of plant-derived compounds?
So regulatory, that means regulatory challenges means that will be in a different sector. Actually, so for clinical translation, as I am belong to a health science campus, so our pharmacy college is located to associated with Amrita Hospital. So what we do for our PhD students or PG students, we work on a particular molecule. If it is of a translational potential, we need to have a kind of positive aspect from the clinician. So what we do, even if we complete the entire preclinical aspects, sometimes clinician may not be in a kind of manner, they won't accept our molecule. So I feel more than the regulatory approval. So what we have to do, we have to convince the clinician. That is the major challenge we are facing. So once the clinician is convinced, so they will allow us to give our product for clinical trial. So they can start the clinical trial and everything. So the thing I am discussing about with the Q-Biomed is different actually. So once we have patented the molecule and they felt it is a very promising molecule, they bought that molecule from us, from our parent institute, and they started. So they are no worry at all. So here our struggle, no, we have to convince the clinician to get approval for clinical trial.
Okay, ma'am. So the Q-Biomed company take care of the regulatory challenges and all of
complete take care. So our only thing is we have to make it into a productive molecule. We have to convince the company like this will work.
Right.
If it works, no worry at all. That remaining thing they will take care of it. Only we can receive that money. That's all.
Thank you so much, ma'am. The next one is, ma'am, how can the bioavailability and delivery mechanisms of phytomedicine be optimized for better clinical outcomes?
So bioavailability is always a issue exclusively for this plant medicine. So even if you talk the Taxol or Taxol, they also not well, like above 50% of bioavailable. No. So for any molecules, it's coming in a kind of natural way, like it has a bioavailability issue. So only thing is we need to have some approaches. So as I mentioned, we have to take help from pharmaceutical chemistry background or chemistry background, make it into some kind of derivatization, as we seen with the capothecin, podophylotaxin. So they made some derivatization based on enhancing the bioavailability or decreasing the toxicity. So in that part, we need to have a collaborator. So or else, what we can do, the formulation aspect, like Taxol
Right.
has a formulation, liposomal formulation is available. So like that we have to make, and like will take time actually, because nobody will believe. So if we give a formulation which will produce a 10 times bioavailability than the parent molecule, so only when some company or has to approach us to do that further study, then only. So because these all need a huge funding for that. At least we can make
Right.
it into a particular preliminary study. So after that, somebody has to take care of that. Then only, or else, so everything is went into a kind of what no use at all.
Right, ma'am.
Okay.
The ma'am, last question is, ma'am, what are the future directions do you see for the field of translational phytomedicine, particularly in the context of cancer research and treatment?
So, so as I showed you in the slide, more than 50% of the drugs available for cancer research is derived from plant only. But what we feel, no, we are not exploring our traditional medicine much, because maybe we feel it will take much time if we are finding something in the dark, or it's kind of difficulties are there. Even if in India we have a number of institute exclusively working on this particular aspect, so nobody come up with a very good kind of the translational phytomedicine. So maybe due to these kind of timing, like is a big problem. And what I felt that currently with the existing phytomedicine already what like already published, what we can do as a say plant researcher, we have to explore its bioavailability issues. Because one molecule, we talk about, if anybody if I heard many times, if anybody work on this phytomedicine, so they will talk, so what about curcumin? So how many years that works have been going on? So lakhs and lakhs of literatures are available, even if this curcumin is not into a translational status. So only taking an example of curcumin, so blaming all the phytomedicine and keep it aside, actually. So what we have to do, we have to move forward. So as a, if we know the potential of traditional medicine, traditional medicinal plants, we have to make it into a next level, like by not publishing what like in average journal, we have to publish the very good data in high impact
Good.
journal. So because when we see that paper status, most of the time we feel like most of the research based on plants are coming into a low kind of journal.
Right, ma'am.
But maybe the limitation will be like they don't have much fund, they don't have much facility to explore. So what we can do, if we feel like if we work on a particular molecule, feel like it gives very good potential, we have to connect with other people. Like we have to convince them and do a very good research, then only we publish. So the number of papers on phytomedicine with a very good research will impact the society, and they will change the mind, like, okay, phytomedicine has good impact, where it's, so only when we prove that through our publication or patent or other approaches, the people will believe on the system and take forward.
Thank you so much, ma'am, for answering all the questions wonderfully. So here
Thank
we
you.
come to the conclusion of the session, ma'am. So
Okay.
in conclusion, the 59th SPSR webinar on translational phytomedicine for cancer has provided an enlightening and comprehensive exploration of the vital role that phytomedicine plays in the fight against cancer. Our distinguished speakers have shed lights on the latest advancement in laboratory research and the promising strides being made towards clinical applications. Throughout this webinar, we have delved into the intricate processes of discovering, validating, and translating plant-derived compounds into effective cancer therapies. The discussions have underscored the importance of multidisciplinary approach, integrating insights from pharmacognosy, pharmacology, oncology, and clinical research to pave the way for novel treatment strategies. Dr. Lekshmi Nath's presentation on the groundbreaking research and clinical translation of Utrocyte B has been particularly inspiring. Her work exemplifies how rigorous scientific investigations can lead to the development of innovative treatments that hold the potential to significantly improve patient outcomes. As we conclude, it is evident that the journey from bench to besides is both challenging and rewarding. Continued collaboration among researchers, clinicians, and industry partners is crucial to overcoming the hurdles and accelerating the delivery of safe and effective phytomedicine-based therapies to patients. Thank you to our esteemed speaker and participants for their valuable insights and contributions. We are deeply thankful to Dr. Shashi Alok, the National President of SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks for your significant input. We are truly thankful for the contribution of our keynote speaker, Dr. Lekshmi Nath, whose expertise provided us with a valuable understanding of translational phytomedicine for cancer, bridging laboratory research and clinical application. We also acknowledge our convener, Mrs. Monika Sabharwal, for her critical role in organizing this event. Our gratitude extends to all the participants for their joint effort, which has expanded our knowledge and encouraged cooperative learning. Your combined efforts have made this webinar successful, and we anticipate further collaboration in enhancing pharmaceutical sciences and research. We are grateful for your significant involvement in the 59th SPSR webinar. Your engaged participation and input have been essential, and we eagerly anticipate collaborating on upcoming initiatives aimed at enhancing healthcare and well-being for everyone. Thank you once again for your participation, and I look forward to seeing you at our future webinars. Keep innovating, keep writing, and keep pushing the boundaries of scientific discovery. Join us for our upcoming 60th SPSR webinar on animal models and neuropsychiatric disorder research, building the bridges. To register for the webinar, please see the registration link in the description box. Have a wonderful day ahead. Thank you so much, ma'am.
Thank you. Thank you.